Good
Hello.
afternoon.
Good afternoon.
Good evening. Good morning.
Good night.
Some of you might be attending asynchronously. So,
That's right.
hello in the future. Are we okay?
Do you think anyone's watching us from the past?
Wow.
Okay. How are you?
Wait, we were just there.
Yeah.
We made it.
We made it.
We're time travelers.
Yeah.
We only go one direction.
How are you? We hope you're okay. We're okay.
We're a solid okay.
We're
Like
just fine. Everything's fine. Where's my coffee?
fine.
We're fine.
We're fine.
Okay. No, we're doing okay. Yeah, and we hope you are too. And if you're not, some of you have gotten in touch. That's fantastic, and thank you. If you need to get in touch, get in touch. The owls are changing, so 1A has become a very happy owl.
I got some Beats headphones there.
Yeah? Oh, look at that. That's good. I'm wearing a hat. And am I listening to your heart? With my stethoscope? I like it. Okay, really good. So
This is all we're doing.
yeah,
Now,
that's it.
we're going to quantify something. And we're going to talk about the visualization of quantification and how truths and lies can be told.
That's wonderful. Oh, we lost our owls. Whoever put that box there, if you wouldn't mind deleting, that would be really helpful. Thank you. Awesome. Thank you. Yay. Okay, so quantification, data visualization. Even in this slide, we have collected data. That's what we're going to be talking about today. And as we collect data, we would like you to not only help us, but reflect on the process, because that's kind of, for today, way more important than the conclusions. For now, let's just hold on to that idea, though. The process is important. We have to critically reflect on the process of collecting data so that we can determine whether it's usable.
The process is the pudding. Isn't
Valid.
that what Marshall McLuhan said?
No.
It was something like that.
Not at all like that.
He used
really.
alliteration. The process is the pudding.
It was the medium is the message,
Right.
and
Well,
in many cases.
Yes, and the medium is the pudding.
So
that's why I think
actually,
about that.
that's really smart, because if we do apply Marshall McLuhan's medium is the message, the way that we collect the data is the message. It matters. What we include, what we exclude, and so thank you. That's brilliant.
Sometimes I think of things that are clever.
Yeah.
Sometimes I say them. Sometimes
Okay.
I say them in spite of them not being clever.
Thank you so much, and thank you for your drawing. And 5A seems to be a leader, although 4C, also quite popular. I'm kind of like, I'm getting the 4A vibe, like kind of thing. But anyway.
I empathize with 5B sometimes.
Yeah. Okay. Oh, and 1C also quite popular. Yeah.
3B.
Yeah. Ooh.
No, sorry, 2B.
2B or not to be?
2C.
Okay.
I don't understand the grade. Not 2B.
Not 2B. The
It's
other
2C.
2B. Okay.
Aye, there's
Good.
the rub.
There we go.
We're
Good.
in that sleep of death.
Okay.
What dreams may come.
Okay. Have we screen captured this one?
We can get it from the video.
It's really pretty. Okay, cool. I'm going to clear this. And for today, if we can practice controlled stamping, because we're going to need you to share your thoughts using stamps. So what we're going to do is we're going to say things like, okay, hold off, don't stamp. Just hover over your answer, and then when we say 3, we'll count to 3, everyone stamp. That way you don't influence somebody else's answer if you're all stamping at the same time. We will try. Some of us will not be able to hold off, or some of us will accidentally tap. That's fine, but let's try generally to follow that.
Controlled stamping.
Controlled stamping today. Okay.
Like stomp.
Stomp. Okay. And so we're going to delete all, because we have a record of it, because it's beautiful. We don't throw these things out. Okay. Just a little reminder, we're not really going to take this up right now, but we are going to ask you to build on this bit of homework to kind of dive into the zebra mussel life cycle. Notice that there isn't this fish host that's part of it, so no parasitism associated with it. And to think a little bit about what the advantages and disadvantages are compared to the glochidial life cycle with the fish host. And to build on that homework, for Monday or the near future, with snow days getting in the way of all of these things, we'll do our best to kind of catch up, of course. But what we would like you to do is to very specifically now think about one advantage and one disadvantage of the glochidia larval strategy, one advantage and one disadvantage of the veliger larval strategy, okay? And what we'll do is we'll give you the opportunity either next class or the class after to share your answers on a Google spreadsheet for everyone to kind of see, okay? So if we all think of one, then hopefully we'll get like 6 or 7 of, some really solid answers. Okay, so that's all I wanted to say about that, because we're gonna do some stuff today.
We've got kind of two, this is one of my favorite classes.
Yeah.
I love this.
Yay, okay.
Isn't this one of the best named papers ever? Harry Potter and How to Display Data Badly.
Okay, so we're going to talk a little bit about visualizing data, collecting data. We're going to kind of rip apart some aspects of the scientific method to kind of probe a little bit about how we as humans interact, interfere, or engage with the scientific method. And by doing that, to kind of just give you a little warm-up to get your brains into the space that we're hoping it will be comfortable in, we're going to talk about how to not engage with data, if that's okay, just to kind of warm you up a little bit. So this is where we would like you to do the controlled stamping. We're going to do a little activity, but first, just to kind of warm your hearts a little bit, kind of to think back.
Oh, Lord. I
To
don't know
not
if this
even
is
the
warming my
last
heart.
time we saw bad data, but maybe the best time.
Yeah. You remember this, Sharpie Gate?
Yeah.
This is where Noah said the hurricane was going to go,
Or, yeah.
here,
Yeah.
because reasons.
Because reasons. I don't even know. But yeah, so don't do this. Don't add a Sharpie onto data to support a mistake that you made. It did actually have quite an effect, though, quite a cost. That comment that Trump said about how the hurricane was going to go further west than it actually was even modeled to go had a big cost.
Mm-hmm.
There were all sorts of emergency programs that were initiated as a result of that comment and all of these things. And so we have to be super careful when we share data, when we use data to support policy or action or things like this. And whoever's putting horns on there, I think it was Maha, and Michelle, I think, is part of it. Very good. Very good. Okay. So clearly badly displayed data because they weren't even data to begin with.
There's also consequences in terms of if you are the scientist, the government federal scientist working on that model, you now have had your boss amend it publicly. And where do you stand? And how do you speak truth to power?
Yep, exactly.
Which is kind of one of the jobs.
Yeah. Okay, good. Now controlled stamping. So thank you for the annotations.
Yep, so let's clear those.
But let's
hover.
Just hover with your stamp. Okay, so what we're going to do is we're going to put up some data or a graph, some visualizations. We're going to ask you what's wrong with them. We'll provide you with a series of options to stamp on, but what we'd like you to do is to just hover over with a stamp, and when we say 3, so I'll count down, I'll count up, then please do click what you think your answer is. Okay? So here we have a graph. COVID-19 cases in the U.S.
Early on, take a look at
Don't stamp yet, please, but please just kind of hover over where you think the main problem is, or there are probably multiple problems, that's fine. And we'll go in 1, 2, 3.
Awesome, and thank you for preserving mostly
I'm sorry.
the
There we go. Yeah.
the label of that box.
Yeah, it gets pretty popular. Okay. Very, very good. Okay. Good. Some of you have clicked other things. The type of graph, it's not bad.
No, it's fine.
I see where you're going with that, though, especially if it was going to be daily cases. If it was cumulative cases, it would probably be okay, but I see where you're going there. Fraudulent presentation. Yeah. I wonder about that too. And certainly these are problems with the axes and the intervals between the different points on the axis. Definitely a problem. So that's really good.
Properly displayed in
okay.
In a second.
Yeah. So what I'll do is I'll clear here. And hover. Here's if we kind of fix it up, the axes anyway. Here's what it would look like. So a bit of a different slope, a different story going on, and certainly you might make different conclusions, right?
And I think back to when this was posted in late March of 2020, when some of the discussion in the world was things aren't going, it's not accelerating. We have one case, tomorrow we'll have 0. We have 10 cases, tomorrow we'll have 0. And it is unfortunately not a characteristic of exponential growth that it goes from 10 to 0, it goes from 10 to 100.
That's right. Okay, really good. Okay.
Well done, you.
Well done. Okay, next one. Are you ready? Let's hover.
I'm ready.
I'm ready.
Let's hover.
Okay. And here we go. This will give you a little bit longer for this one because I feel like it's a little trickier. So just take a look. You don't need to know what the y-axis means, but basically what it means, I feel like you should talk about what it means.
You don't need to know.
You don't need to know. Basically, what it means is sort of the ability it's talking about the genes, right? The DNA. Basically it's sort of like the strength of the bonds, basically. But anyway. Oh, you're already stamping. Okay, go ahead and stamp then. Some suggesting some axes problems. Most of them are going for the pink one.
Yeah, typograph and labeling seems to be
typograph and labeling.
Yep.
Okay. Yep. Monkey. Yeah. Okay. Super good. So type of graph, let's focus on type of graph. If you want to pop into the chat, what type of graph would be more appropriate in this particular case?
Bar is one suggestion.
Yeah, bar's not bad. Bar's not
question
bad.
Mark? Bar?
Yeah, I feel almost like a box plot. What do you think? Just because...
Do you guys know what a box plot is?
Yeah, so a bar is definitely better. And the reason why something that is...
Great point, Irigala.
It implies,
the connection implies this led to this led to this, and that is not the story.
That's not the story. So one of your comments is that it implies that humans evolved from monkeys. It can imply that, and even more ridiculously, what this graph implies, to me anyway, is that an individual can change species. So let me read it to you according to what it says here by using this connected line. It says that as something goes from being a rhesus monkey to a gibbon to a chimpanzee to a human, it changes its like splice site strength, right? Like it shouldn't be connected in any way. A bar graph is better. I think a box plot is best. But certainly having those connected lines, not a good thing, not a good idea. Okay, we're gonna clear, and then
this is one of
on
my
3.
favorite
Hold off,
ones.
please, hold off. Take a look at this one.
Hover.
Just hover, and on 1, 2, 3, go ahead.
Cool.
Very cool.
We have a trimodal distribution. We have a tripod.
Okay. So the one we'll focus on for this one would be the incorrect inference. So that's the orange second from the bottom. You know, these are real data. These are real things that are highly correlated, but not at all causal, at least not directly causal. You might anticipate that in a country that consumes more chocolate, they would also have a higher socioeconomic status, which means that they would probably fund more science, which means that they might then have more Nobel Prizes. But that's like 3 graphs away to get there in terms of causal relationships. So, so certainly there are some problems associated with putting these two things on the same graph, opposite each other as axes.
Yeah, certainly there, as you can see in the label at the bottom in the title of the figure, they're not saying that we love this graph because of how you might have it incorrectly used, but in this case, they are saying correlation. They're not saying cause. The error is in how you infer the cause from that.
That's right.
Because chocolate is not the mechanism from which Nobel Prizes emerge.
But wouldn't it be amazing?
Wouldn't it be amazing? Wouldn't it be nice
if
we could eat some chocolate and then go
Great.
collect the Nobel Prize? All right, next one is touchy. So this was a Chiron and a display on Fox News talking about the fact in the Unfortunate Truth, or in Al Gore's documentary, he described in 2007 that the Arctic would be ice-free. And then Fox News is like, Al Gore is lying because look, in 2012 there was this little ice, and now in 2014 there is this much ice. Al Gore is a liar. Now to sports. What's the problem? How do you lie, or how do you present data with graphs?
Next.
Let's go to the next one. So this is actually the data, and if you're interested in kind of harvesting this kind of data, there's a link there at the bottom. But take a look at 4 different images there, and what I want you to do is hover.
Hover.
And now use the stamp tool. Show me the 2 images that were compared on the Fox News report. You are all over it.
Yep, very good.
And as you're stamping, so what's the problem? The problem is you're saying it's time and it's season.
Yeah.
It's like Al Gore never said summers would be colder than winters, or winters would be warmer than summers. It's like,
yeah,
it's bananas. So in terms of the other kind of categories that we had earlier, this is, I would say this is pretty close. This is selective. This is data harvesting to tell a story. So this isn't
fraudulent representation.
This is fraudulent representation. This isn't evidence-based decision making. This is decision-based evidence making.
Very good. Yeah. Okay.
Which is explained in the next slide. Come on.
Yeah. Okay. So obviously some things are a bit easier for us to kind of piece together and to be able to critically evaluate. Other things, not so much. And so we want to kind of dive in a little bit now and I think a little bit critically about biology, biological data, and to kind of open up some of the uncertainty associated with it.
Can I interrupt from the chat before we go on to active variation? The question is the stuff like this on the topic test? In a sense, yes.
Yeah.
Because the numeracy and the literacy of looking at a graph and pulling out what the story is, and then down the road for you making a graph, making a figure, and telling a story. Those are intimately important jobs of a scientist in any branch, whether it's human health, animal health, whatever it is.
Yeah.
In business, it's an important job. And so you have to be able to look at these images and receive the story. And, of course, if there's going to be fraudulent presentation, you have to be able to identify when you're being sold that fraud.
That's right. So in terms of like prep for the topic test, we've already done some together.
Yeah.
What we like to do during these times together is to study for the topic test, or to learn together, right?
There's another quote.
There's another question.
Yeah.
So to get you into that mindset, into that sort of way of thinking. So really do your best, and I see all of you, well, a lot of you are doing your best to engage and to get your brain into that space. That's all you need to do.
And so that ties back, before we jump into the human measurement of the phenotypic variation in human section, is that the question was, are a lot of my notes going to come from the pre-lecture readings? And so yeah, they are.
Sure.
Because what we're trying to do here is we've moved a lot of that dumping of stuff, the presentation of the context, to the pre-lecture section. That's X number of hours that we talked about in terms of your 10 hours a week on this class. And we're trying, as Dr. Jacob said, to engage with you here in this little Marceau box that we all share. Hold on. Wait
We're
a second.
Okay, cool. So, let's think a little bit about it with respect to biological data.
Basketball.
That makes you think of basketball?
Yeah.
Okay.
Some of you fans of the Raptors at your birth, you might remember from when you were 1, 2, and 3 months old that the Toronto Raptors had a player named Muggsy Bogues. And Muggsy Bogues started his career in the late 80s there with a team that no longer exists from Washington, where he was the shortest player in NBA history, and he was a teammate of the tallest player in NBA history. Nearly a human in between them in height, 71 centimeters difference.
That's right. So Manute Bol was from Sudan and was the tallest player, or still is the tallest player. Yeah, he passed away due to complications associated with being extremely tall. But was fantastic at putting the ball in the scoring goals. No, what do you call it?
Points. And blocks. An enormous number of blocks.
And not so good at running quickly down the court, but that was kind of what Muggsy did. And for 1 year, they actually played together. Yeah. And it was really quite a sight to see.
And you're right, Muggsy himself had a cameo in the original Space Jam.
Yeah. Manute was also a really active person fighting for social justice for his country, and so really both of them actually. Muggsy Bogues too does lots of social justice stuff. No?
Yeah, he does. He's from a rather impoverished portion of Baltimore, I believe, and so a lot of his is about equity and representation and lifting up, sports for lifting up.
Yeah, cool. Okay, so big difference in a single trait that we're hoping you're going to get to in a second, and that is height, right? Big difference between these two, and certainly across humans, that's kind of a big thing that differs among all of us, right?
Yes.
Yes. Okay, so let's consider the trait then of height, and take a little bit of a look at how that height trait is distributed across different humans. And we can visualize these data in many ways. This is a way that was done a very long time ago from the quality of that photo. But imagine then that those wearing white are female and those wearing black are male. And then we distribute them by their different heights with 1-inch intervals. And we create a bit of kind of a curve, right? This like bell curve. That bell curve has a very specific name. It's called a normal distribution. So when we talk about normal distributions, we are referring specifically to this bell curve in which the mean is right in the center at the very peak of the curve, right? But there is variation that exists within the population that frequency of those expressions of variation diminish as we get closer to the ends, okay?
Gaussian is also correct.
Very good.
This is not named after Dr. Normal.
That's right. So a normal distribution is a very deliberate and specific piece of jargon in biology or in statistics. Okay, though, if we look at this, is this really the best way to visualize the data? Right? What do we think? Like, just looking at it, I mean, if we're not interested in sex differences, then sure, it's probably fine. But then if we are interested in sex differences, if we're not interested in sex differences, why are we getting people to wear different color clothing?
They're probably part of the band.
And really what it does is allows us to see the difference associated in the distribution of those people wearing black, but not so much for those people wearing white. Yeah. If we wanted to perhaps appreciate the differences within the population associated with another trait like biological sex, first of all, we might actually represent more than just 2 biological sexes. But if we were just going to focus on 2 biological sexes, we might prefer to do it this way so that we can see if there really is a difference in the mean, for example. Okay? So there are like multiple better ways of representing data, and it really depends on what it is that you want the person looking at the data to get from it or to focus on. Okay.
So there's a whole bunch. I remember from the start of at the start of the course when we were asking about what major you're in, and there were a couple of you that were in visual arts or fine arts, and visually representing data, there is an art to it. There are basic rules that artists learn about in terms of the use of space and the use of and how to tell a story visually, and scientists could do really well from learning from that.
Okay, so what we'd like you to do is something. So you'll need a ruler, and if you don't have a ruler, you can go to the one that we put on the welcome screen. Do you want to pop that link into the chat for anyone who still needs it? You do have to tell them what size of monitor you're using, but just kind of guesstimate it. You know, if it's 10 inches or 15 inches or 18 inches, I don't know, I've got like a 30 something inch here, just so that it can scale to the right size so that you get like a true to scale ruler. Otherwise, use one that you've got lying around the house. And what we'd like you to do is to measure the lengths of your ring on your left hand, your ring finger, which is this one here, and then your index finger, which is the one that you point with. Okay, so just go ahead. If you're rude, and measure those. We do note that about 250 of you have probably already done this.
Well, you've watched us describe how to do it.
That's right.
Yeah.
So what we'll do is we'll get sorted out now to start collecting the data from you, so that those that have already done it can pop in their data first. But for those who haven't, no worries, just go ahead and do that. We want those two measurements. Okay.
So to harvest this data from 1,000 of you and to come into this little room without passing us envelopes underneath the door, which was what we were going to do first of all, what we're going to do is have you segregate by your surname, the first letter in your surname. I've just posted in the chat these Google spreadsheets, links associated with surnames from A to C, D to F, G to I, J to L, blah, 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 all the way to Z. So find your surname, and then when you have your measurements, find the appropriate column for you and drop them in.
Yeah.
I see some of you arriving already.
Okay.
Hello.
So we're gonna divide it up. Those of you we have divided by two sexes, female and male. So when you get into your spreadsheet, you'll see where we would like you to pop in those numbers. And just go ahead and do your best.
Yeah. So
ask us any questions in the chat if you have any.
If you identify as male, find the male column. If you identify as female, find the female column.
Great. And remember to think about what you're doing. We're gonna ask you questions.
Are you Mad-Eye Moody? Do you remember Mad-Eye Moody? It's a Potter reference. Constant vigilance. Remember to think about what you're doing.
Always.
Oh, always?
No.
Left foot, right foot, left foot.
Somebody's asking us to go faster.
iFleet, sorry, how do we join? I'm going to say it out loud and then I'll post it again. Scroll back down in the chat and you'll find a link. That's how you join the Google spreadsheet.
Yep.
And the links are below. I'll repost them.
Repost them because a whole bunch of you just joined in the chat, which is delightful. Thank you. A lot goes on in the chat. I know it might be distracting though, so avoid it if you have to. Nothing like, we don't like put learning materials in there or anything like that.
Oh, that's a good. I saw that some of you are putting in units. Don't have to put the units in the cell.
Numbers.
This is actually a great lesson, little micro lesson about recording data. Only the number. Never put the units, so don't put MM.
Thank you. Somebody just deleted their units.
Amazing.
Amazing. Really good.
Only the number.
Okay, and then if you have any questions, pop them in the chat as well. And then I'm going to have to go and refresh, right?
Yeah, do you want to take it off shirt because something just happened.
Okay. That looks fantastic. We'll just give you another minute or so. we lost this one.
No. It'll come back.
Will it?
Yeah.
I can't do refresh though. Ah, there we go. Should we work with this?
Sure.
Maybe another second. This one.
Oh my! I was on the edge of my seat because I didn't know if it was going to work.
Yeah.
It worked.
It worked.
1,000 people.
1,000 people on Google Docs. Amazing. Are we almost ready?
Yes.
Okay.
And we're sharing the screen. That's the dairy bush.
Oh yeah.
There you go.
Isn't that good?
Snowy, snowy.
Yeah. Can we get...
Where's the chat? I can't access the chat.
Oh.
So if you go back to course link, I'm just talking to people who've annotated the screen.
Yeah.
And spoke about not being able to access the chat on the home page on your dashboard for course link. All the titles up at the top here. Chat is right over here. Click on that and you'll see different chats for different days. And click on the one that has today's date, 21/01/19.
Is this one better? Or no?
No.
Okay. Okay, I'm going to do one final update. I don't know it. And wonderful. Okay. We're just going to make sure that we have the graphs all generated. Great. Okay. Ready?
People are making observations about the length of their fingers.
Yeah. Okay. Thank you so much for all of your contributions here.
And we will still see them if you continue to add. If there was a little bit of traffic, that's fine. Keep adding them, but we'll continue with the presentation.
Okay. There we go. Wonderful. Okay, perfect. So we just in like 4 minutes collected quite a bit of data, and that's kind of a super fantastic power that we have in this class with all of these students to be able to do this, and so we're very grateful.
So take a look at those y-axes.
A look.
Take one of the things I already see, and this is true in most years in biology, take a look at the difference in scale between the left-hand side of the screen and the right-hand side of the screen.
Right. Yep. Okay. Why don't we all kind of do this for the next few minutes? Take a look at these 4 graphs and write down some things that you notice, either some conclusions, some differences. Pop them in the chat.
I'm going to use the other...
Yeah, we can. Okay. Oh yeah, if you want, we can do that directly. Okay.
I think is it the next?
Yeah, okay. Okay, so we'll pop in some Google spreadsheets for you to do that, because actually the first column is exactly that. What patterns, what differences do you see? So that we'll just jump straight to that. I wanted to do one thing with them before though. Okay, perfect. So just take a look at all of these things. Notice differences, similarities. See if you can come up with like some big conclusions, and there'll be like multiple things that you might notice. The one thing that I wanted to kind of do all together right now is to take a look at the distributions of the data. So of these, which one looks most like a normal distribution? Go ahead and just circle or stamp or whatever right on the screen. Okay. Really good.
Yeah.
Okay. Yes. Very clear, right? And it's balanced on either side as it diminishes. It's not skewed to one side or another side, right? Okay, perfect. To figure out whether thank you, Malik, yes. To figure out whether or not the index length for males is normal or not, there are some statistical analyses that you can do that will tell you whether it's close enough to a normal distribution to be normal or not quite normal enough, right? And certainly the same thing can be said for the index finger length for the female population or the female subgroup, right? The question is for ring length of males. Do we know for sure whether this is a normal distribution or not? Can we know for sure? Zoe is saying no. Jonah is saying no. No. A bunch of people are agreeing with you. Yes. Okay. Good. Yeah. You can't say for sure, right? Oh, somebody's saying why in the chat. But the reason why you can't know for sure is because the visualization fails us. Where does the visualization fail us? In appreciating the distribution of the data for the male ring finger length. There we go. Ahmad, very good. Very good. Yeah, Kiana, exactly. Exactly. So if you take a look down here, we've organized, the scale's not long enough. Thank you. Yes, Callum, greater than 90. Exactly, exactly. That's right. Here, for all of the others, we can see the distribution of the data. We can appreciate whether it's normal or not. But because our visualization fails us in representing the longer finger lengths and sort of just bunching them all together, we can't say for sure or definitively that the male ring length is not normally distributed. It's a failure of our visualization if what we want to do is appreciate the distribution. If we don't need to appreciate the distribution, then it doesn't matter. But in terms of figuring out whether it's normally distributed, it's probably entirely possible that it is, right? It's probable that those data are also normally distributed, but our visualization fails us in being able to appreciate it, okay? So now we can do your spreadsheets.
So we have some questions, some 4 questions that are displayed on the screen there, and they are posted in the chat. And
here.
Yeah. So what we're asking you to do is to revisit those spreadsheets, the Google document where you were, not where you entered the data. There's another sheet where these questions are there displayed, and you have a chance to kind of, and I've seen some of you already hop in and start entering your thoughts as to why this might be some problems. Yes. Somebody wrote this sheet? Yes.
Wonderful. So this is really studying. Like for all of the topic tests, this is kind of what we're going to do most of. We're going to give you data. We're going to ask you to come up with conclusions. This isn't the only time that we're going to do this together in class, but it's kind of the longest time that we're going to spend really like focusing on how we interpret what we can conclude, okay? So please go ahead and give it a try.
Yeah, and the stuff that I'm seeing, you guys are getting this. There's people talking about data limited to students involuntarily participating, blah, blah. Yep, those are important things.
Go.
How representative.
Yeah. So we'll give you just a few minutes, and then we'll totally discuss. We need to make a very important sort of note in all of this before we move on. So we'll just give you a few minutes, though.
This is being recorded, right and left.
Yeah.
Thanks to Durla for pointing out that the link to D to F is the same as the link to A to C in that posting.
Cool.
We're juggling a lot of spreadsheets here.
Yeah.
Okay, so there's a request in the chat to go back to the graphs.
Absolutely.
They have those questions in the chat. They can see them.
Sorry. Yes, that makes a ton of sense. Of course we can go back.
Hello. Salut Daniel, ça va? Tu as un câlin pour Lily?
Amuse-toi, bonhomme, ok? Non, je t'adore. Oh.
Merci pour la câlin.
Okay. How's it going? What are they saying? Oh, all sorts of things.
Aha.
Yep.
everything there.
Yep. Super good.
Yeah.
Mm-hmm. Love that one.
Da.
Yep.
Yeah.
Mm-hmm. Yep. Wonderful.
Sure.
Super good. Okay. Yeah.
Put your right hand up in the air, or your left hand, whichever you want to. Pat yourself on the back, on the shoulder, on the top of the head. Well done, you.
Well done.
You have generated a large data set.
Like a really big one?
Like larger than has been published in Nature.
Okay, so let's kind of run through some of these things because you've shared some really important things.
Yeah.
The big thing that I feel it's really important to kind of put out there in the world, especially for people who are training to be scientists or who already are, is this whole idea about methods. And the more that we kind of dive into the methods and the more that we practice the methods, the more we realize how precarious those methods can be because they are done by humans. And humans have all sorts of problems. And also computers have all sorts of problems in terms of biases and things like this. And so, thinking a little bit about who we excluded in this particular activity is really critical to point out. So we excluded people without left hands. We excluded people without ring fingers. And we also excluded people who didn't have rulers. We excluded people who are non-binary, both either in terms of sex or in terms of gender. And we excluded a whole bunch of other people too. And it's really critical to understand that, especially if you're not somebody who is usually excluded from these types of protocols. Just to kind of tell you a little bit about what that might look like. It might look like a drop-down menu on a survey that you just can't get past because you're not there. But it can also look like you submitted all of your data thinking that you were going to be represented within the study, only to read a few weeks later that because non-binary representation was less than 3%, we eliminated all of those data. And I'm kind of sick and tired of not being represented. Like, I have strong opinions. I want my opinions represented.
Wait, what?
So you're either, as if you're a non-binary person, and like I can kind of speak to this a little bit more, specifically, but certainly if you're a disabled person, you're either not allowed to participate or you're not included in the conclusions. So they either don't allow you to give your opinions and your data, or they don't care about it. And I'm not sure which one's worse. And so this is something that I think, especially as we go into the human sciences, something that we need to rethink a little bit. It's very nice to have your drop-down menu that's inclusive, but unless you're going to use those data, I'm not sure that that's anything more than just virtue signaling. And it's important in science to kind of recognize that.
And you think scientists would be good at categorizing and thinking about data like this, but they're not. There's all sorts of publications where people try and use meta-analyses, for example, to determine gender based on names and then make enormous conclusions from that. And it's just like, no, why? No. As someone who grew up named Sandy, it's like there's a lot of that's gonna not work.
And somebody who went for 15 years as Sean, that would be like oh my goodness. Yeah. So, humans are complex, and I think most of you, especially in the winter group of 1070, are really interested in human science, and I think it's an important thing to consider.
Say it louder for people at the back.
Yeah.
Yeah, and there's some other people in the chat and in the spreadsheets have had other thoughts about, like, we're not using the same methods. We give you a very cursory description of how to measure.
We didn't train you.
Were we doing it the same way? How standardized was our measurement?
Very
we
good.
Didn't know where to measure from. We gave you kind of some drawings, but...
Yep. Yeah. Oh, and somebody's saying, actually, my right hand is much smaller. Yeah, there's all sorts of nuance to these things, right? And the same type of nuance. You might then go and say, well, I'm going to use, molecular tools to be able to,
As if that was bias-free.
It's like, not bias-free. It's all biased, right? How much do we know about female spiders?
We know next to nothing about female spiders because all of human history, how we have identified spiders is by identifying and naming male spiders.
Right.
You think that's a problem?
Who wants to become a female spider biologist?
Female spiders are not rare. You find them literally as frequently as you do males.
Okay.
And male spiders aren't even identifiable as males until they're adults. So, de facto, you always find, most of what you find, you can't identify because of the gender bias inherent to the taxonomic process.
That's right. And it turns out that female birds do sing. So these things are important, and there's tons of opportunity to take an old idea and reevaluate it from a more inclusive and equitable lens, both in terms of human biology, but also in terms of like all the other biology out there in the world.
Female spider justice, I like it. So, and then thinking about female spider justice, we have to say salutations, because we have to think about E.B. White and Charlotte.
That's right.
And her web.
Okay, so.
But it gets worse.
It gets worse. So this study was published in Nature, the leading journal in science. Like, this is the one that like everybody goes to for like the truth. And some guy named Mark Breedlove decided to go and measure the finger lengths of a whole bunch of people.
Not as many as we just did.
We did way more just now.
Yeah.
But then Mark put the data on a terrible graph. This graph is virtually indiscernible.
And you're looking at it going, this is old, this is 2000, this is the year 2000, but it's like, we certainly knew how to graph better than this in the year 2000.
We could do better than this. What he found was a correlation between the ratio of finger lengths and sexual orientation. Now, this is not meant for you to look at your finger and go, dude, I'm really straight.
Oh, no.
No, not at all. It's again a problem or issue of correlation and causation, right? That I think we probably should unpack, although we don't have to, because a whole bunch of people already did unpack it.
Oh, there's a lot of sliding.
There's a lot of unpacking.
There's a lot of unpacking of Mark going on in the.
Wonderful.
Mark measures 5 people's hands. Good enough.
Yeah. Okay. So tons of skepticism, tons of critical reflection that are going on associated with these data.
But it's a whole industry.
It really is.
So this is not something that has gone away. That study has been cited more than 600 times.
Yeah.
And there's a whole industry because, as we just showed, and you just participated in, it's easy to measure.
Right.
And there's a whole bunch of people that are unpacking and using it because they're like, well, I'm going to infer fetal levels of androgen.
Yeah.
By measuring the fingers of adult Olympic athletes.
Think about all of the other variables that might contribute to those types of measurements, right? Including nutrition, including access to healthcare, including, right? Okay. Yeah. So other people have pushed back on this. And if you're at all interested in this world, in this conversation, in this discussion, to kind of practice a little bit your critical thinking associated with not only the biases that we introduce in our methods, but also the ones in interpreting data, it's kind of a fun space to be in.
There's a couple of people that are commenting that this came up in their elementary schools.
Amazing.
This, right, the 2D40 ratio.
Wow. Did they also tell you what your orientation was?
Wow.
Whew. Okay, so how does this apply to mussels?
Yeah, remember mussels.
Remember mussels? Yeah. Think about it a little bit, right? We introduced the idea of intraspecific variation. So that is that individuals have variation in a trait like height or finger length. Right? Same with mussels in terms of their size or shape or color or number of ridges or number of byssal threads or all of those things, right? Intraspecific variation. That this variation can be caused, at the level of phenotype, and also it could be as a result of differences at the genotype. And natural selection has a role to play on variation. It selects variation that contributes to more reproductive success. and that all sorts of other evolutionary mechanisms can get in the way of that, and so it isn't these like perfect linear relationships that say if you have this variation you will be selected for in this way and you will have this many offspring, because there's a lot of like interplay and mixing going on in the environment as well. So that's kind of a quick rundown, but if you'd like to kind of take what we talked about and then fill in your explanations for how this might fit within these bullet points, that might be a really good way of kind of reviewing from this class. Yeah? And with that, we'll say thank you so much. Your enthusiasm is just
well done.
Such a pleasure. So thank you all very much. And we'll be here for an hour if you need us.
We're going to turn off and then turn back on, but be kind to yourself, and when you do that, be kind to others.
Take care.
Stop the share.
I'll stop the recording.